Let's get started. Welcome everybody to the 11 o'clock session with Dr. Vockley to discuss preclinical treatment studies in FAOD. We'll take questions at the end of the presentation. You can submit your questions again using the Q&A at the bottom. So back again with us today to share more of his wisdom is Dr. Jerry Vockley. Dr. Vockley is internationally recognized as a leader in the field of inborn errors and metabolism. His current research focuses on mitochondrial energy metabolism, novel therapies for discovery of fatty acid oxidation and amino acid metabolism, and population genetics of the plain communities in the United States. He has published over 270 peer-reviewed scholarly articles, is the principal investigator of 4 NIH grants and a co-investigator on 7 others. He has an active clinical research program and participates in and consults on multiple gene therapy trials. Dr. Vockley has served on numerous national and international scientific boards, including the Advisory Committee to the Secretary of Health and Human Services on heritable disorders in newborn and children, where he was chair of the Technology Committee. He is co-chair of the International Network of Fatty Acid Oxidation Research and Therapy, also known as INFORM. He also serves as chair of the Pennsylvania State Newborn Screening Advisory Committee and the American College of Medical Genetics Therapeutics Committee. He is a past president of the International Organizing Committee for the International Congress of Inborn Errors of Metabolism and the Society for Inherited Metabolic Disorders. He is also co-founder and editor of the North American Metabolic Academy. Please join me in welcoming Dr. Jerry Vockley.
All right, thanks, Kyra, and sorry for some of the technical issues here. It seems that we have a lot to learn about video conferencing, but we're here. I'm pleased to come back today to talk to you. We talked a little bit yesterday about the transitions occurring in the field of fatty acid oxidation disorders, and this morning I'm going to focus on some transitions that I hope will be happening in the future, talking about preclinical treatment. And I also like to title this discussion or talk Novel Therapies for Inborn Errors of Fatty Acid Oxidation, a personalized medicine approach. You hear a lot about personalized medicine, this is just another way of saying that while we have had relatively few tools to use in treating these disorders in the past, triheptanoin now gives us a new one in our armamentarium, but there are going to be more coming down the road, and it may well be that some of these are going to work for some diseases and some patients, and others will work for different. So the more tools we have, the better off we are to likely to be able to treat you. So unfortunately, you're still not here. I am. But I'll do a little bit of travelogue for you here through the show, show you some stuff through the talk. Pittsburgh, of course, is the city of 3 rivers, and while this is not an exact representation of the city, in honor of our meeting last year in Amsterdam, I like this representation. This is the Monongahela and the Allegheny Rivers coming together down here at the point to form the Ohio River. And if you were here, you would be able to go down there and grab a boat or a bite and enjoy yourself. This is near where you would have been staying had you come to Pittsburgh in Oakland. The Phipps Conservatory, one of the largest conservatories in the country, and quite beautiful. Just some conflicts of interest, or as I call them, opportunities. And we're going to start way back at the very beginning. What now is, I think, junior high biology. I don't know. It keeps getting younger and younger. And remind you that DNA, that chemical that carries all of our genetic information in every cell, has to be duplicated every time it passes on to a new cell. And sometimes one of those duplications can be an error, and that causes what's called a mutation. And those mutations can lead to genetic disease. The next step in the transfer of information in a cell is what's called DNA transcription, where a copy of that DNA is now made from into a compound called RNA. RNA, all of our genes have extra material in it that isn't used to make proteins. And so that gets spliced out and makes what's called messenger RNA. And that messenger RNA now becomes the blueprint for making proteins. Amino acids get hooked one after another until you get the whole length of the protein made, and that's the enzyme. Those are the things that carry out the chemical reactions of fatty acid oxidation. So when you are looking at this kind of process, you can also think about how how treatments for fatty acid oxidation disorders can be focused at each level. If you want to fix the DNA in a cell, it has to be up here. That's gene therapy. You can provide more protein. That's down here. I don't even have a picture of the protein. But a lot of times things that are the mutations in genes that make proteins will actually allow a protein to be made, but it doesn't fold properly. Proteins are two-dimensional when they come off at this stage because you're adding one amino acid at the next, but they have to be folded into 3 dimensions to work, and sometimes that doesn't work. So there are molecules that can help proteins fold. And there are mRNA. If you can give mRNA for the protein, you can make more of that protein. And we're going to talk about all options in all of these areas. You saw this slide yesterday if you were online. The interaction of the mitochondrial energy pathways. Here, fatty acid oxidation, which can pass fuel directly to the respiratory chain, which makes cellular ATP. That's the fuel that our cells can burn. Or it can send its biochemical product called acetyl-CoA down here into this other cycle called the TCA cycle, which in turn makes more fuel for the electron transport chain. And so all of these areas all of these pathways are worth knowing about, because we can sometimes demonstrate secondary defects in one of them when there's a primary defect in another. And the one that you'll hear about today or throughout this meeting is triheptanoin, which is a fuel for fatty acid oxidation. But it actually, what it does is it fixes a secondary imbalance in the TCA cycle that is a problem, an extra problem when you have a fatty acid oxidation defect, because then instead of just one abnormality, you have 2 that you have to deal with. And then finally, you need to know a little bit about the mitochondria, this bag of enzymes that serves as the battery for the cell, and where fatty acid oxidation is loaded. This is a little tiny structure that you need a microscope to see, and it has 2 coverings called membranes. One out here that just sort of contains everything, and another one inside that's got these funny invaginations that allow the architecture of the mitochondria to be optimized to the needs of the enzymes. And so most of energy metabolism actually down here in these crypts. To make this structure, and here I said you have to be inside, you have to look at a microscope. Here's a very high powered called an electron microscope, and you can see where this comes from. But this isn't actually quite right. If you look at this in 3 dimensions, it looks a little bit more like this. So instead of a pinball machine where the pinball starts up here and goes bing, 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 and comes out down here, it's actually this set of things where you can put a drop of fluid up here, and it can go over this way, it can go over this way, it can come up here, it can go all the way around. And the interesting thing about this configuration here inside the mitochondria is there's a very, very special fat, a lipid called cardiolipin that's necessary for this to happen, for this configuration to occur. And that's going to be of importance in fatty acid oxidation disorders and in a way that was unexpected before we started looking into it. And I'll come back to talk to you about that. And then finally, if you come down to the very, very sub-molecular level, when you look at the proteins involved in fatty acid oxidation, they also take on a very special configuration. It isn't just like one protein's floating around inside the mitochondria and it finds a fat and it does a chemical reaction and lets it go, and that finds the next protein in the cycle. Turns out they're all interacting, so they make this physical complex. Here in this cartoon, the respiratory chain with the proteins of the respiratory chain that we now know both physically and functionally interact with fatty acid oxidation. And if you have a problem with this protein, for example, it could potentially cause this protein to fall apart, the trifunctional protein, and vice versa. And so that gives us other ways to look at trying to think about the treatment of fatty acid oxidation disorders. We're back to Pittsburgh. So unlike that stylized Van Gogh representation of Pittsburgh, here is a photo of the Three Rivers, and it's quite a scenic city. The Allegheny, the Mon, the Ohio going off down to meet the Mississippi. The downtown area here, and Oakland, where the university is back here, which is where you would have been staying. Pittsburgh's a city of neighborhoods. There's dozens of neighborhoods. They all have their own special feel, and it's an old city. It's a lot like Boston in that regard. You can walk down a street and you can see something like this that was built in the 70s and the 1700s, I'm sorry, or the 1800s. Here, looking right downtown into the modern downtown area. So it's quite a nice city to visit. Now, I talked about all of those basic characteristics of energy metabolism. Why am I worried about that? Why do I talk about that? Well, it does have clinical implications. And you can think of it as a house of cards. If you have a problem up here, if this card isn't present, the rest of the house is just fine. No worries about it. But what happens if you got a mistake down here with this card? Well, the whole thing falls apart. And this is something that we now know plays a considerable role in fatty acid oxidation disorders. I like to go just a basic biochemistry here to show you what a fat looks like. Okay, this is what a fat looks like. It's a whole string of these carbon atoms hooked together, and then there's this molecule on the end, which is kind of the business end. This is where all the chemical reactions occur. And when you have fatty acid oxidation occur, you knock two of these carbon units off at a time. That makes acetyl-CoA. And every time you do that, you make fuel for the respiratory chain. Excuse me. When you burn glucose for energy, you make 34 molecules of ATP. That's that cellular currency of energy, the molecule that the cell needs to burn. When you burn this molecule, palmitate, C16, you get 123 molecules of ATP. So you can see that burning fat is a lot more energetic than burning glucose. So it's a good thing. The problem is if you can't do it, if you've got a defect in long-chain fatty acid oxidation, not only don't you make anything, but it turns out to get the whole thing started, you need to spend a couple of molecules of ATP up here. So if you try to, if you have to rely on fat for fuel, you're in a real hole. For long chain fatty acid oxidation, or those, you can still do the medium chain reactions. And so theoretically, you can get about half of this out of the process. But the problem is that when you're using MCT oil, the only thing you're making, MCT oil is even chain carbons, predominantly C8. The only thing you're making if you take 2 carbon units off at a time is this 2 carbon unit, acetyl-CoA. Well, the body shares these chemicals from the TCA cycle with other activities in the body. And if the only thing you're making here is bringing in our even chain carbons, it turns out that some of these actually require odd chain carbons. And so this cycle will run and it'll run and it'll run if you give it C8 until all of a sudden it runs out of those odd chain carbon compounds, and then it stops. So how do you get that? How do you fix that? Well, you bring in, instead of a 2 carbon unit, a 3 carbon unit, odd chain carbons, which fill up the cycle here. And this is why C7 is a better fuel for long chain fatty acid oxidation disorders than C8, because it provides both 2 carbon units and 3 carbon units to balance out the TCA cycle. Now that's in theory. Is it true? Well, yeah, it is true. We've studied a lot of patients with VLCAD deficiency, we can measure all of these intermediates of this TCA cycle here, each of those compounds is shown here. And what you can see is anything below the graph, below this line is too low, and anything above at the line or above the line is normal or increased. And what we have here are 6 patients treated with triheptanoin and 5 patients treated with MCT oil. And for the most part, what you can see is that the individuals who were treated with C8 have lower levels. They're more decreased than those with C7. And it turns out that for the most part, the molecules that are here, there's enough of them to make the TCA cycle work. So that's why we think that C7 is a better fuel for patients with long chain fatty acid oxidation disorders than C8. Now, originally this was going to be a talk about triheptanoin, or it was going to include triheptanoin because it hadn't yet been approved. And I was going to pull these slides out, but I thought, well, you know what? You really ought to understand how the studies went to allow us to allow the FDA to approve triheptanoin. And this is a study that Dr. Gillingham and I did with a grant from the FDA, and some of you participated in this study, where we brought you into the hospital, did a bunch of very fancy testing, and I'm not going to go through all of this, and then either gave you C7 or C8 in a double-blind fashion. That means neither you nor we knew what you were taking. Then brought you back into the hospital 4 months later and did all those tests again. And the most important thing that we found out about this, and one of the things that the FDA was most interested in when they were looking at it for approval, was that triheptanoin allowed better heart function than MCT oil. In this graph, it shows the amount of heart work over time depending on whether you got triheptanone and MCT oil. So the lower you are on this curve, the less work you used. And this was a test that was done on the treadmill over 40 minutes of time. And what you can see is that the triheptanone patients had lower heart rates that whole time, which means they were working less than the patients who just took MCT oil. And Dr. Gillingham had previously shown that if you took MCT oil, you did better than if you just had a carbohydrate load prior to your exercise. So in total, the improvement from carbohydrate to C7 is quite significant. Now, we didn't see a muscle effect here. It was a very short-term study. But this was still, because of cardiomyopathy is such a devastating problem in this disorder, it was good to see that effect. In the Ultragenics trial, we focused more on the clinical side of things rather than looking at the biology or the physiology. This trial was done by taking individuals who were already on treatment with MCT oil and then switching them over to C7 and comparing their clinical picture after they started the C7 with before they were on it. And you could see that of the patients who were part of this study, almost all of them had muscle problems. A couple of them had current cardiomyopathy, and even more of them have had a history of a problem with cardiomyopathy. The disorders were evenly split among CPT2, LCHAD, TFP, and VLCAD. What we did was we just counted the number of clinical events that happened before the switch to C7 and after. And by clinical events, we meant rhabdomyolysis, hypoglycemia, or a problem with developing or worsening of cardiomyopathy. Both in counting the number of events and the number of days related to hospitalization, everything decreased on triheptanone compared to C8. So we essentially eliminated hypoglycemia. We decreased problems related to the heart by about 70%, and rhabdo was the least affected. It dropped by about a third, and the episodes were less frequent and less severe, but rhabdo remains the one problem that is least well addressed by triheptanone. So we'll come back to that in a little bit as we talk about why that might be and ways we might work around that. The drug was safe. The GI symptoms were the biggest problem. And they were very similar to what patients experienced with MCT oil. Most individuals who switch from MCT oil to C7 don't experience anything new, anything different related to the new drug. And everything that was seen in after switch to C7 was at about the same frequency as before C7. And so, as we discussed yesterday, TriHep was approved at the end of last month, and it really is a big. It's a big deal, and I have to call out my children's hospital team because, of course, we were involved in working with triheptanone before Ultragenics came on the scene, and Elizabeth McCracken, one of my senior study coordinators, has been involved in this right from the very beginning, and so she and the rest of the clinical team, Jen Baker, Danielle Black, Angie Lashinsky, and Dr. Georgianne Arnold, are really to be commended for the tremendous work that they've done on our studies, as well as the studies in collaboration with Dr. Gillingham and Ultragenics. So you'll have an opportunity to learn more about Ultragenics as the conference goes on. If you weren't online for the discussion yesterday or for the presentation yesterday by Ultragenics, by all means go to their booth in the exhibitors area and chat with them. Pittsburgh again. The University of Pittsburgh is in Oakland. Here's that downtown area that I showed you before. So the Monongahela River is up here, and the Allegheny River is over here. You can just barely see a little bit of blue of it here, actually. And this is the Children's Hospital. But oh, I'm sorry, no, it's not. That's the VA hospital. We're not. Our Children's Hospital is back here. This is this is the university campus. It's an urban mixed campus. So there are lots of university buildings, but in and amongst this are urban streets with businesses and residential areas. So it's quite a unique campus in that regard. And here, the most recognizable building here, the Cathedral of Learning, or the Tower of Learning, which is a building that's still used for classes. So it's both a recognizable landmark and a functional university facility. All right, let's get back to work. I mentioned that triheptanone helps but doesn't fix the rhabdomyolysis. Why might that be? Well, one of the possibilities is that in any disease that causes muscle inflammation, you can have rhabdomyolysis. And I thought, well, what is it? Maybe we're seeing something with inflammation in our patients that we just haven't recognized. The usual markers of inflammation in patients that you might be familiar with, a SED rate or something like that, are normal in patients even when they're sick. So we went and we looked at a variety of different things. The first thing we did was to look at the chemicals that we know influence signaling for inflammation. Why might that be an issue? Well, in fatty acid oxidation disorders, in long-chain fatty acid oxidation disorders, there are two things that you have that are a problem. Think of a water pipe with a block. You can have you have two things you have to worry about. You have a deficiency of what's supposed to be coming out of the other end, water, or in the case of a fatty acid oxidation disorder, energy, but you also have a backup behind that block that can cause problems. In the case of a water pipe, flooding in your basement maybe, but in the case of fatty acid oxidation disorders, all the fat that's getting broken down and hits a wall, can't be metabolized, hangs around, and the body can do other things with it. One of the other things it can do with it is that these fats are used in the body to make other compounds, complex fats or complex lipids that it uses for a variety of things, including signaling. And so here's just an example. I'm not even going to try to get you to pronounce these molecules. Sphingomyelin, a couple of different kinds of sphingomyelin, a phosphatidylcholine molecule, another complex long chain fat, and they are elevated in patients with long-chain fatty acid oxidation disorders. Now, this one is actually, these are the odd chain molecules, so they're higher in C7 than they are in C8 treatment. But if you look at the even chain ones, it's flipped around. So whether you're using C7, C8 for treatment, you accumulate these molecules. And what does that do? Well, these signaling chemicals induce an inflammatory response where that makes molecules called cytokines. And these cytokines are more subtle signals of inflammation than some of the things that we were routinely used to look at. And this is what's called a heat map. And I'm not going to worry too much about the details on this, just to tell you that all of these molecules over here on the left are things that induce inflammation and potentially could increase rhabdomyolysis. We measured them in 16 of our patients. It turns out that half of these were on MCT oil and half of them were on C7. So there was no real difference. And anything on this heat map other than green is abnormal. So you can see that patients with VLCAD deficiency have a lot of these things that are abnormal. And if the red, they're really abnormal. And this is a potential problem. Equally importantly, what you can see is that each of these lines on this graph represent a hospital admission of one of our patients. And every time that patient was admitted to the hospital, we could see increases in these cytokines. And so it looks like there is an underlying inflammatory process going on in our patients, and it needs, so we need to try to think about how to treat that, or how to deal with that as part of our treatment. One of the other, and there are a variety of anti-inflammatory compounds that are out there that are used for diseases that you might have heard about, autoimmune diseases, things like lupus or MS, where the body is attacking itself. And so we can look at that, at those compounds, and see whether they help out in patients with rhabdo. The other thing that I mentioned early was that fatty acid oxidation and respiratory chain have to interact well for all of this to work. And when it doesn't, the respiratory chain generates something called reactive oxygen. Now, there's a lot of lay literature about reactive oxygen. The most important thing for you to know is it's usually not good for you. A little bit is normal, a lot is bad. And here you can see one of our patients. We were just looking in skin cells from that patient, and that patient has a huge elevation of these reactive oxygen species, and this chemical that can cause damage. And this is both a cause and a result of inflammation. So it kind of sets up a bad feedback loop that leads to more and more damage over time. Well, we have an antioxidant that just has a name, a number and letter name, JP4, that gets targeted specifically to mitochondria. All the other antioxidants that you can go to GNC or Amazon and find, type in antioxidant, you'll find all kinds of compounds. None of them get to the mitochondria. And when we treat cells from patients with fatty acid oxidation disorders, it does nothing. But this compound reduces that reactive oxygen. And so we're very interested in trying to move forward on a clinical trial using this compound in our patients with fatty acid oxidation who are still having trouble with rhabdo, and see if this can make a difference in their clinical symptoms. That's not just true for VLCAD deficiency. You can see here cells from a patient with LCHAD, the common LCHAD mutation, have very, very high reactive oxygen species. Here's a patient with TFP deficiency, also an increase in reactive oxygen, but it's not uniform. Here's a different patient with TFP, and this individual has normal reactive oxygen species. So I come back to my title of this, personalized medicine, and we're going to have to do studies like this on patients to try to understand the effect of their fatty acid oxidation defect on their body, so that we can best treat you. If we have 10 years from now, if we're lucky enough to have a dozen different treatments, which one is going to be best for you? Well, we could just try by trial and error, but this would be a better way to do it. So as I said, we're doing more studies on that JP4 molecule. Hopefully, somewhere down the road in the not too distant future, we'll be able to develop a clinical trial for that. Now, the other thing that we're looking at are molecules that help abnormal proteins fold properly. Remember, I told you that proteins start out as a two-dimensional one amino acid attached on the other. Well, then once that two-dimensional structure or string of beads, if you will, is done, it has to fold up in the proper configuration. So it's like taking a string of beads that's now, you hold it out like this and you've got a long string, and then you have to start tying it in exactly the right kind of knots to get it in the right configuration, as opposed to just dropping it on the table and have it turn into this blob on the table. One of the ways that we're doing that is with a chemical called trimetazine, we can treat cells with this drug, which can help any fatty acid oxidation protein fold properly. And you can see here that starting here down at no trimetazine and up here at a higher concentration, we can make more VLCAD protein fold in a cell with a mutation, and we can make more TFP fold in a cell. So another drug that we're looking for to be able to do a clinical trial on. The Reneo drug, which I mentioned yesterday, and they're also out in the exhibitors hall, so you can go talk to them, works by a different mechanism. If you go back to my basic biology scheme, DNA has to be made into RNA. So every protein involved in energy metabolism has its own gene. And the body has developed a way of coordinating how much of those genes are turned on. And when your body needs more energy, it can send out a single signal that says, make more of all the energy proteins. And this is the signal that it sends out it interacts with a piece of DNA that's called a promoter. And in the case of energy metabolism, one of the strongest promoters is called PPAR delta. And if you have a chemical that interacts with a PPAR delta promoter, it can induce all the energy genes. Well, this Reneo drug, as I mentioned, it doesn't have a name yet. It's just got a number, REN001. Is a very, very strong inducer of gene expression. And so here you can see in one patient with VLCAD deficiency, we start treating with the Reneo drug, and we about double the VLCAD activity. Here, slightly lower concentration, but the same thing. We increase the activity, and we've shown this is also true with the genes for trifunctional protein. So this is the trial that's ongoing now, and we've been on pause because of COVID for the last few months. But hopefully, by the time we get started, by the time we get to September, October at the latest, this will open back up, and we'll have a little bit more details about who's eligible for this trial in the talk tomorrow with Dr. Gillingham on clinical trials. For long chain defects, the last molecule I'll talk to you about is something that interacts with this fat that I told you about earlier on, cardiolipin. Cardiolipin is kind of it both allows that funky configuration of the mitochondrial membrane that lets it bend to make those those special places for enzymes to sit, but it also kind of serves as a glue that holds the enzymes of the respiratory chain together. And since the fatty acid oxidation proteins are interacting here, we thought, well, maybe it's necessary for those as well. We have a drug that can help. This is a molecule, this cardiolipin, that's very easily damaged by that reactive oxygen that I mentioned. And so maybe cardiolipin's abnormal in patients with long-chain fatty acid oxidation disorders, and maybe treating them with a drug that stabilizes cardiolipin will help. So here's a patient with LCHAD deficiency. I showed you this before. Cells from that patient have a lot of reactive oxygen species. But when we treat them with a cardiolipin binding peptide, something that helps fix the cardiolipin in that cell, it comes back down to normal. So we're working with another company called RQ8 Therapeutics. They have this peptide, and we're looking to bring that to a clinical trial sometime in the not too distant future. Going back to our basic biology, mRNA makes protein. So if you've got an mRNA that doesn't work, or it makes the wrong protein, can we give you mRNA back? Well, there's a company, you might have heard of them, Moderna. They're out in front of the race to make a COVID vaccine, but they're also working with us to make mRNA treatments for VLCAD and MCAD. And here, what you can see is if we inject VLCAD mRNA into either a cell line or a mouse that's deficient into it, we get a huge bump in VLCAD activity. The same thing holds true for MCAD. And in fact, we can normalize the activity. The problem is right now this is liver specific. So we're not sure it's ready to be a treatment for VLCAD because so much of the problems are related to heart and muscle. So this is a kind of something to keep in mind for the future, and we're going to be working on it. I had a question yesterday about our MCAD therapy, and this is another one of those therapies that's designed to help proteins fold. The common variant, the common mutation in MCAD is a change that turns a lysine into a glutamine, glutamate rather, two different amino acids, and causes the MCAD protein not to fold properly. And this is something I discovered almost 30 years ago when I was a fellow, and it just kind of sat there for a while. But it turns out that there's an FDA-approved drug called phenylbutyrate that is metabolized by MCAD to its active form. And I thought, well, maybe if MCAD is metabolizing this, it might also be able to use it as a chaperonin. And I've shown this data at a couple of these conferences now. It turns out that if we take white blood cells from a patient with MCAD deficiency, you can see there's almost no activity there, and we treat it with this drug, we essentially normalize the activity. So we think that this is going to be a drug that will be very beneficial in the younger age group where problems with MCAD deficiency are most pronounced. And then even for older individuals, when you're sick or if you have ongoing symptoms, you might be able to use this as a sick day medication. And we're doing a clinical trial where we bring patients into the hospital and we measure chemicals that we know are abnormal in MCAD deficiency. Here are some molecules called acylglycines. And what you can see here is that of the 3 patients that we've studied thus far, all 3 of them have had a drop down to almost normal of the acylglycine that was abnormal in them. It tends to differ from patient to patient. So I really think this is going to work. In fact, we've shown that it works in these patients, and we're looking for funding to be able to do the clinical trial to get this approved by the FDA. Fortunately, since this is an approved drug already, it's much easier to get that approved for a new disease than it is to go from scratch. So I'll keep knocking on doors and windows and car windows, whatever it takes to find somebody ultimately who's able to fund this trial. I'm going to stop there. I mentioned bring some of my collaborators over the years on some of these studies here, and stop and show you my last picture of Pittsburgh. And this is a lovely piece of architecture called Falling Water. It's built by Frank Lloyd Wright back in the 1940s for a prominent Pittsburgh family, and it's really it's one of the a world architectural landmark. It's about 45 miles south of Pittsburgh, and if you ever do get to visit Pittsburgh, I highly recommend going to visit. I think it's one of the most spectacular pieces of residential architecture that I've ever seen. So let me stop sharing here. And I know we started a little late. It's noon. Kyra, do you want to do some questions, or do you want to put those off to one of the discussion rooms?
Hey, Kyra had to jump off, so I missed her now. Hey, Dr. Vockley. There's a couple questions in the chat. You just want to answer it? We can go through those real quick, and then we can jump over to the breakout sessions.
Sure.
People can continue those. Okay.
Let's see. I've got one that says, are Mito cocktails recommended? The answer is there's very little evidence that they work in mitochondrial disease, and therefore I don't use them in fatty acid oxidation disorders. Some folks like to use some coenzyme Q10, and it acts as an antioxidant, but it's not a very good one. It might help stabilize those respiratory chains, the complexes I talked about, but they've never been studied, and I've never been convinced that they make any difference. Next question: when should you use a carnitine supplement versus an over-the-counter version versus a prescription version? Tough question. I always try to use the prescription version if I can get it approved through a patient's drug plan, because the quality control is there. There's so many examples of supplements that have been taken off the shelf, tested, and showed to either have very little or none of what was supposed to be in the bottle. And so I don't like to use them. If you can't get it approved by your insurance, and you feel like you need to use it, then yes, you can go get an over-the-counter supplement. See if it's someone that will give you the chemical analysis sheet of the batch that they're selling you. So you can go online to a company, and sometimes they post those analytical sheets, or sometimes you can ask them, can you show me your sheet? I want to know there's actually carnitine in your bottle before I use it. So something to do. Best way to avoid the GI side effects of triheptanoin. The same things you use with MCT oil is the easiest way to say that. With the trial, we were stuck using it straight up. That was just the way the trial was designed. Now that it's approved, you can do any of the things that you do with MCT oil. You can mix it in with salad dressing. You can sprinkle it on pasta. You can mix it in with yogurt. You can bake it into brownies. So lots of options there. And in fact, Dr. Gillingham is going to be putting together with her team a cookbook on that. So keep your eyes open for that. CPT2, daughter with CPT2 was on the C7 trial. Will she be allowed to be followed for the next 10 years? Yes. If you want to sign up for that study that Ultragenics was talking about yesterday, she will most certainly be able to be studied. In fact, I would encourage it. We need to know what happens to our patients either on or off triheptanoin. And this is the best way to do it. Ultragenics is providing significant funding for us to understand this, so it's a good way to go. That will either be through the same study site that you went to, or in some cases they're going to be opening up study sites elsewhere to try to make it more convenient for folks. Let's see. When do you think the clinical trial will open up to individuals under 18? I assume that's the question on Renea, which right now is over 18. That'll be the next phase. So hopefully next year. And not too far along in next year. Question about how the LCHAD studies relate to the VLCAD studies that I pointed to. I use VLCAD, LCHAD sort of interchangeably in my presentation. Many of them behave. Many of the studies are the same on both. Some are different. It most definitely comes back to that personalized medicine concept that I introduced at the very beginning. One of the best ways to do this is to look directly at your cells or your child's cells and do these tests that we do in the laboratory. Right now, I'm the only place that does that, and I can't do it on everybody, but eventually as we move these things forward, hopefully we'll be able to do that on a more regular basis. Oh, what can we do as parents and advocates to help push the funding for Ravicti through for MCAD? You got to do the same thing as me. Bang on doors, windows, streets, pots, pans, anything you can. Companies that make this drug, Horizon, funded that first study that I did, but then kind of shrugged their shoulders and said, our priorities have changed. We're not interested anymore. Call them up. Yell at them, harangue them. They make both Ravicti and and a different version of it called Bufenil. There's another company that's making a phenylbutyrate a drug called Acer, you may know I get calls all the time from folks who are looking for projects. These are venture capitalists with lots of money to spend, and they say, we want to try something. If you know somebody like that, have them call me. It actually would be a relatively inexpensive trial from the standpoint of pharmaceutical trials to get done. Now, when I say relatively inexpensively, keep in mind, I'm still talking probably about, and I'm making this number up, probably a couple $1 million. But it's not the 30 or 50 million that Ultragenics invested in triheptanoin. So just to put it in context for you. MCT safe over the counter. The problem with over-the-counter MCT oil it is not all C8. It is a mixture of C8, C10, C12, and sometimes even C14. So you have to know what's in the preparation you're using. Same thing with that I mentioned previously with over-the-counter carnitine. If you can go to the website of that company and find out what was in there, you will, and it's all C8, should be okay. But this is why when we talk to insurers about pharmaceutical grade MCT oil, it is 100% C8, and that's what you want. So I'm going to stop here. I've got to get off to the next my next breakout room, but I'll be hanging around for much of the meeting, and I'll try to look at your maybe Jeannie, you can send me those questions and I can answer them, and you can post them. Just send them to me by email. I can send them to you and you can post them up, or you can grab me in one of the chat rooms and ask about them. Okay.
Yeah, we can sure do that. Thank you so much, Dr. Vockley, for your time.
Thank you all, and
As we all know,
I'm off to another chat room, so I'm not exactly sure what it is, but
I
I'll
just
see
sent
some
you the
of you
link to
there.
it. All right,
Bye bye.
everyone. So we're heading into our afternoon break now. So please be sure to visit the lounge. In the lounge are the disease or diagnosis specific groups. Dr. Vockley will be there. You can continue the Q&A there. Or Reneo Pharmaceuticals also has a Q&A going on. So there are many options to choose from during this break time. Also, don't forget to visit the exhibit hall where you can find our sponsors and vendors. And then we'll be back at the auditorium for the afternoon presentation starting at 1 o'clock. So thank you all so much.